Welcome everybody to this H3D Foundation webinar. We're delighted to have Dr. Kirandeep Samby with us today, talking about library design and how that drives drug discovery. Dr. Samby gave this presentation or something similar earlier this year at an in-person workshop, and we were delighted to share this with a broader network. So I'm going to read the bio as by way of introduction, and then I'm going to hand over straight away to Kirandeep. Dr. Kirandeep Samby is a drug discovery scientist and R&D leader with over 20 years of experience across the pharmaceutical industry, public research institutes, and global health organizations. A PhD graduate from Delhi University, she began her career as a medicinal chemist at Ranbaxy and Daiichi Sankyo before moving to CSIR-IMTech and Medicines for Malaria Venture. She is currently a distinguished scientist, director, in communicable diseases at Johnson & Johnson. Her expertise spans infectious diseases, antimicrobial resistance, and inflammatory disorders. She is co-inventor of multiple clinical and preclinical candidates across therapeutic areas. Alongside her scientific contributions, she has led cross-regional discovery programs, built strategic alliances, and advised startups advancing innovation in antimicrobial resistance. Combining deep scientific expertise with strategic vision, Dr. Samby offers unique insights into translating discovery research into impactful global health solutions. Kiran, we're so thrilled to have you, and thank you so much for giving us this time and expertise. You're also a beloved friend of our network, particularly as a mentor in both our cohorts most recently. And so we're so thrilled to have you. So thanks everyone for joining. There'll be a Q&A at the end as always, and I'm going to hand straight over to you, Kiran. Thank you.
Thanks, Nicola. And, I have loved coming back because, as we were talking, it gives you, it gives you a different kind of energy and brings you to, a different level to, probably to answer some questions. And I understand that it's not going to be, may not be that interactive as we have face-to-face seminars, but I'm happy to have any questions after the presentation and, happy to interact with people if they have specific questions even after this session. Okay, having said so, I'll move to my presentation. And I think most important thing to understand is, what we are screening and why we are screening. And this presentation is going to, mainly cover different aspects of library designs. I have not specifically kept that very technical because, what I realize is that there would be diverse audience. But at the end of the presentation, what the purpose is going to be that why library design matters in modern discovery. And, what we have been using traditionally and what all the changes, that has been coming to science in I would say last 10 years or 15 years brought different screening approaches and a few recent case studies that appear to be changing the game of how we think and how we screen libraries. And this is what I was saying, that what and how. Probably most of us in this forum, at some point of time, be it a biologist or a chemist, would have seen, to start a new project. So how do I start a new project, right? If I need a novel chemical matter, of course, if it is a me too, it's not actually first in class or it's a best in class molecule, then, there are different ways of looking at it. But these are, we are always at crossroads that what and how, what should I screen? Because there are so many options. As you see on the left-hand side, either the screenings to be used or what to screen or how to screen, right? So what I hope is that by the end of this presentation, and the discussion, following this presentation, we would have some clarity that how we should look at the samples to be taken up for screening, and do we, and what kind of assay systems we should be looking at. So I think, when I say about what kind of assay systems should we be looking at, it would be more focused towards the infectious diseases, but then, they can be easily extrapolated to non-communicable disease areas as well. Before we actually enter into what kind of technological changes have happened and what a library design or an attribute should look like, I'll just take a step back uh, and see, what high throughput screening is all about and how it, evolved, because it also gives an insight into, right, what we see today was not always not always it was like that. And probably 10 years down the line, it is going to be very different from how we see it today. Uh, so if you, you know, if I can, kind of explain this timeline, I would like to see that majority of the work started with, uh, screening of natural product libraries. So basically, and that's another topic that we'll discuss towards the end of the presentation. So, because those were, fermentation extracts, which were screened. And as you see on this slide, there were, couple of thousand essays that could be run in 2 weeks. But then as drug discovery, started to pick up, and the areas, of discovery ventured beyond the infectious diseases into non-infectious diseases, and evolution of biology, there were a lot of changes that were happening. So you see that initially in, late 1980s or 1990s, how what high throughput screening actually meant was basically doing a 96 well assay, either a phenotypic or a target-based screen with a large volume that we call today 50 to 100 microliter, but at that point of time, it was a small volume, right? And how, all these learnings and development of the concept of screening, compounds in parallel in 96 well assays or 384 well assays transformed how we, screen compounds for their various ADME properties like microsomal stability or even for that matter, permeability or cytotoxicity assays. And in those, I would say 20 to 25 years of discovery of this high throughput screening, it was more about that the compounds were segregated, the compounds were accumulated based on, whatever the compounds were synthesized in the projects. And so those formed the basis of the libraries that existed with various drug companies. And in early 90s, the scientists started going beyond those things, where combinatorial libraries of something of the scale of 200 to 500,000 compounds were generated. Few simple reactions, which were, maybe amide coupling, nucleophilic substitution reactions. So, which were quick to do and easy to purify, and that resulted in large libraries but with very limited diversity. And at one point of time, the physical chemical properties were so bad that the concept of, are you synthesizing sand or bricks, came into fame, because those compounds were highly hydrophobic and had poor physicochemical properties. So the compounds lacked solubility, they were non-specific promiscuous inhibitors. And in early 2000s, when actually those combinatorial, and pharma companies realized that screening even hundreds and thousands of compounds is not yielding the kind of results that were expected. So when those cleanup of libraries actually, led to removal of almost 50% of the compounds in some libraries. So, but having said so, all was not that bad. Because it also led to discovery of uh, few drugs. Uh, and this table clearly shows that this is an early impact. So all the HTS that were run in the 1990s led to the FDA approvals of these compounds, which are marketed even today. And as you would see, again, very limited diversity in the sense of, the targets per se, most of them kinases and proteases and some GPCRs. But yes, so all this work that had gone in 90s and, 80s and 90s actually has created a platform of developing the things further. So if we look at, to explain the clear impact of the table that you see over here, there are two case studies which I have picked up. And, I realized that both of them are for, basically antiviral compounds. So if we look at on the left-hand side, the chemokine receptor antagonist by Pfizer. So HTS of 500,000 compounds were done, and it took almost 10 years till Marveric was approved in 2007. So the compound, the discovery started with this, which had no cellular activity and a weak agonist activity. So almost 4,000 compounds were synthesized to dial out the undesired activities and identify potent antiviral compounds in this example. On the other hand, there was an example of thrombopoietin receptor agonist by GSK. If the medicinal chemist probably in the cohort can easily identify, oh, these are certain functional groups like bisphenols and azo compounds that we would avoid in today's time. We'll never go down that lane. But at that time, yes, a cell-based luciferase reporter screen assay was done around 250,000 compounds. An initial hit was actually called the nuisance compound. But since there was a robust SAR and the biology could be deciphered, it led to discovery of L-thrombobutyric. Right. So these two case studies actually set an example that how the scientists have been thinking in the field. So in today's time, right? So, and both of these are examples of hit identification to moving to compounds in clinic and finally FDA approval. But is it that library screening matters only in hit identification for a novel target, or is it that something it goes beyond that? And the answer is that if we look at why screening matters, that screening matters at all stages of discovery. Early stage discovery programs, for which we called hit identification, when there are no valid starting chemical material to work on. So for the first in class targets, to get a starting point, or a workable chemical space. We have seen all the time in oncology for kinases where inhibitors for mutated type targets are already, all have to be identified so that the resistance mechanism can be addressed. The compounds, probably the prior compounds already have a very well-defined crystal structure. The mutation is known that why resistance is being caused. And with those modifications, you start looking for new chemical matter, and library screen becomes a go-to, I would say, a go-to strategy. And then, if we take a step back and come back to, infectious diseases, and, if we are looking at not specifically at a target, but specifically at some, pathway blocking, then phenotypic screens also become, important, and compounds with unique biological activity can be identified. And this is again, an example that I'll give later in my slides. So not only early discovery stages is when we are looking at it, but also from hit to lead or beyond. And of course, for a chemist or a project leader who is running a project or a program, they are always looking for a differentiated backup scaffold so that, issues with pharmacology or toxicological issues as the project progresses further could be, addressed if the need arises. And it is, so what we call is that it is to enhance the drug-like properties to minimize the downstream failure of with those compounds. So I think it is not about screening, how big the library is that is used for screening, but it is about what the library contains and what you are screening against. So smarter the library, the better the chance of finding workable chemical spaces. So if we look at the high-level design strategies, that how these libraries are designed, of course, one and the most commonly available is a general library, which could be bespoke, I mean, made on demand, or it is readily available through commercial vendors and pharma companies. And I'm sure that all of you on this platform are aware of these general libraries. Then there can be focused libraries where um, the libraries are enriched with compounds that target specific pathways. And again, if we go back around 10, 15 years ago, kinases were the hot stuff. So there were a lot of libraries that were synthesized only for kinases. Most of the pharma companies, actually started their combinatorial chemistry efforts to focus on those kinases. And that also, what we call as an orthosteric ligands. And then, there are GPCR libraries. And as the technological advances have happened, there has been a lot of focus on peptides and macrocycles, as well as protein-protein inhibitors, which have been thought to be difficult to target pathways. Then there are fragment libraries, which contains smaller molecules to probe specific binding interaction. And since, as the name suggests, these are small molecules which can be expanded or grown based on the structural information that is generated. There are a lot of natural product libraries coming into being and utilizing all plant microbes and marine organisms. And the reason is, again, we'll have a separate session to discuss about why natural products is regaining that focus, which was there around 50 or 60 years ago. Initially, the work on natural products was slowed down because of the complex organic synthesis associated with it and low yields. But now, again, the way of looking at those natural products is becoming totally different. And then again, there are a few commercial libraries, either in the form of FDA-approved compound libraries or, low-packed libraries, which are library of pharmacologically active compounds, which are smaller sets, already screened. And I will show you a classic example that how, development of various assay systems have positively impacted use of these libraries. DNA-encoded libraries and large virtual libraries are the hot topics these days. Again, as DNA-encoded libraries is something that we'll discuss later in the presentation. What is very important to keep in mind is chemical diversity versus relevance. Like I said, a couple of slides ago, that are we screening bigger or smarter? So we could have a diverse library, but if it is not relevant, so we may, it may lead to actually a very low, probability of success. So biological relevance is very important to get, translational value of these libraries screening. And these days, most of the time, hybrid approaches are being used where diverse libraries are being, selected, but they are filtered for the desirable properties or based on the information that is known for the target. So there are a few things that have to be kept in mind for the key evaluation metrics. One, of course, is diversity. And like we have been talking in past couple of slides that the diverse library increases the likelihood of finding active compounds against various targets. And the typical ways of checking the diversity is the Tanimoto Similarity Index, which quantifies the structural similarity based on the fingerprints. And also to keep in mind is the fraction of sp3, because we do not want to be in the space where you have, highly hydrophobic compounds which are not developable. And all this can be done with the help of chemical space visualization tools like principal component analysis, that can help you keep a track on the diversity of the libraries that are being used. The second is the quality, whether the compounds are pure, stable. And when we talk about stable, it is about the chemical stability of the compounds and what would be the general in vitro admit properties of those compounds. Again, both for reliable screening results and to, and to dial out any promiscuous inhibitors which may result because of the instability of compounds is something to keep in mind over here. So when we talk about representativeness, it is important that, we see that there is a diversity, and diversity is, slightly different from representativeness in the sense that when we talk about representativeness, we see that there is an adequate chemical space that is represented in the library, and it is not, and the library is not biased towards known scaffolds or chemotypes, which would be probable active compounds for that target. And for that, clustering analysis is done to see that how diverse the library is. And of course, there is targeted library design to see that the biology and the DMPK properties or drug-like properties of the compounds are taken into account. So at the same time, there has to be a predictive value to assess that how well the library can predict the biological activity. And a classic example is that if you are looking at the protein-protein, disruptors, protein-protein interaction inhibitors or disruptors, you would not be looking at compounds, typical kinase hitters, or kinase inhibitors, I would say, or a very because the for protein-protein interaction inhibitors, the pockets are shallow, so the structure of the compounds have to be something which have to be kept in mind while you are looking at the predictive value of these compounds. So when we talk about what to screen, of course we have seen that we take diverse compound libraries. And if we are, and when do we take those diverse compound libraries? Are we exploring novel targets? Are those first in class where there's no initial chemical matter known? We do not know. We do not have sufficient information about the target structure per se. So that would be, that would be the target, or that would be the screen when you would, take a very diverse compound library to see that what kind of chemical matter actually inhibits or or is a agonist or an antagonist for that target. Right. The second thing when you pick up the diverse compound library is when we are looking for hits with unique mechanism of action. So basically, we are trying to do new phenotypic screens, right? More predictive of how the compounds would behave in vivo or in the human physiological conditions. And of course, first-in-class discoveries would need diverse chemical scaffolds so that we are not restricting ourselves, or we are not having a library bias towards ourselves. The only challenge that maybe, that can be identified with this thing is that hits may be less target specific. So that would, increase the list of false positives. So that is something to take into account. And that is why when these assays are done, there are always counter screens that are carried out with those compounds. And specific enrichment, it could be, there are two ways of looking at specific enrichment. First is that we have already explored a novel target, and you already know that what kind of scaffolds or what kind of chemotypes are active. So if there is a specific enrichment more similar to those scaffolds or chemotypes, or when the target is already well characterized, there are structural insights available, so those things can be used. And what we are saying about a hybrid library is like what I simply just explained, that you have a diverse set of compounds that are screened. You have already identified hits, and then there is a coarse diverse library with smaller target or family enriched subsets that actually helps in identifying of hits. Stage gated approach or computational approaches that we are following using AI ML to predict the compound target interaction with the discovery of AlphaFold models and things like that. So as we saw on the first slide that, screening could be done both in traditional as well as innovative way. Most traditional, when we say that, there are high throughput screens, phenotypic or simple target-based screens, or we do a natural product screening or fragment-based screening. But with, again, development of so many assay platforms, there are organismal systems that, that are being used for screening. DNA encoded library is one example. Virtual screening where billions of compounds are docked if the structural information of the target is available, followed by wet screening. And then there are, AI for de novo design where based on the structural information of the ligand, millions of compounds can be designed and then docked, and then wet screening can happen. So there are a lot of innovative strategies that are coming up, and taking away the traditional role of high throughput screens. So I think, and with this, and with these technology enablers, there are ways how we can screen smarter, and there are ways like large ultra dockings that are happening, make on demand virtual libraries, generative AI spaces, and human and machine integration happening to screen the compounds in a smart way. It's not necessary to screen billions of compounds in the wet lab, but there are ways of filtering out those compounds to a reasonable amount to screen those compounds. So this is this is a classic example of compound library, how the compound libraries have evolved over a period of time. So initially, like when we were, looking at the first few slides, the compound libraries were of the size of 10 to the power 3 or 10 to the power 4. But today, if we look at the stock collections of the molecules that are already available on the vendors' shelves, they are something between, millions of compounds, and there are more than 100 chemical vendors that are available. Then on the other hand, there are also on-demand databases and spaces that they're available. They are all virtual, but they are readily synthesizable. And as you see on the right-hand side of the slide, there are more than 120,000 building blocks that are available, and more than 180 reactions can be done at different areas of diversification, and on-demand molecules can be generated. So basically, there are 10 to the power 23 to 10 to the power 60 molecules that are actually possible, which can be synthesized virtually, and then followed by, the various computational filtrations can be, managed into a more reasonable number. So one thing that we have been talking about is, structure-based virtual screening in last couple of slides. I mean, I have said these things. So what does, structure-based virtual screening would mean? I think, with availability of various 3D structures, either what is already available in PDBs or what are available with AlphaFold, right? Predictive structures. So that as one vertical, and then growth of virtual chemical space and possibility of make on demand libraries on another vertical, along with the computational methods and accessible computing power that that the academics or groups have across the world today, has led to development of various techniques for structure-based virtual screening. One is of course, when we are talking about millions of compounds, it is brute force ultra large docking. So it can use up to 1 billion compounds for discovery of novel ligands. But one thing to keep in mind is that it is very, very resource intensive, and you should have those kind of computational tools as well as computing power to run those kind of docking protocols, right? Another is fragment-based method where out of the full molecules, the based on the chemical scaffolds or the fragments are extracted, and these selected fragments are either co-crystallized with your target, or from the docking predictions, they are expanded and followed by again docking and wet screening. The third way of looking at is machine learning surrogates, that a subset is selected and used for training of models, and then again predicting through databases. So this is a, sorry. This is a classical case of, I would say, ultra large library docking. And as you see that there were around 150 million lead-like molecules which were docked using dock 3.7 in the crystal structures of MT1 and MT2 to identify MT1 selective ligands. And these molecules were selected from the Zinc database. Now, when you are screening, docking so many molecules, so top 300,000 molecules were clustered by topological similarity. And the compounds which were very similar to MT1 and MT2, were removed. And the best scoring around 10,000 clusters were actually visually inspected. So here it is where, when we talk about AI and human interaction to understand the key interaction and select only 40 compounds for synthesis. So, you see that talking of hundreds and millions of compounds, zeroing down, filtering it down to a few 100,000 compounds, and then there are 10,000 compounds which were, manually eyeballed to select 40 compounds for synthesis to identify MT1 selective inhibitors. So, till now we have focused only on chemistry, that yes, these kind of libraries are available. These should be the parameters that we should look in the libraries for picking them up for screening. But, having said so, the other aspect is how to screen, right? And those technological enablers for large scale screening are, either microfluidic systems, which actually, brings down the requirement of your compound to few microliters, and then is also 3D cell culturing, high content screening, gene editing, and of course one of the hottest topics, like I mentioned, was DNA encoded library. So in which the DNA is used as a barcode so that compound libraries can be screened within a single microplate. So this is what the purpose of DEL screening is. So if you look at this, that there are billions of small molecules in a single test tube, I would say. It is a bespoke library that is constructed based on the information that is available for the target. These compounds are screened in the test tube to and then followed by sequencing and decoding of these compounds. The hit compounds or the binders are identified. And once the binders are identified, the decoding helps in identification of those compounds. But what, in past 10 or 15 years, these DNA encoded libraries have been able to deliver is compounds with novel protein conformations. New binding sites have been helped in identification of new binding sites, and of course, novel protein ligand interactions. And this this slides actually shows that how DEL screening has diversified into a way that that the scientists are looking at. So if you look at some of these examples, the optimized structures are not very different from the optimized structures on the right are not very different from what was identified from the DEL hit screen, and currently they are in different phases of development. So it is complement to traditional high throughput screening, because you yourself see that it is not about synthesizing screening arrays and arrays of compounds. It is synthesizing billions of compounds in one go. It provides a broad chemical space to work on. And most of the time, there are truncations that happen on the initial identified DEL hits to improve the potential and physicochemical properties. And in the past couple of years, improvement in DNA compatible transformations and various transition metal-promoted reactions or radical-based reactions have helped in generation of such libraries. Having said so, there are certain challenges which still need to be addressed so that, in future, it can be used more appropriately. Of course, the most common challenge is that there is so much of background noise because we are looking at an affinity and we are not looking at the inhibition potential, right? So, and larger the libraries, there, could lead to higher rate of false negatives because how they are truncated or based on data interpretation. But there is a lot of work ongoing in this area, how to minimize uh, the false negatives. And then, of course, there is use of machine learning that is happening to determine, what are the potential binders from the background noise and how do how can informatic tools can expand the improved physicochemical space within the Dell libraries. Sometimes, most of the times where Dell libraries have been used are, have been used where the target protein has, basically an extracellular target because the entry is an issue. But recently, there are, two sites-based Dell screenings that are happening because it helps in overcoming the challenge of the requirement of purified target protein, as well as the cell permeability issue that may happen with these Dell screens. So I think, I'll not go into this natural product, which ones of those are, but because I'm sure that, most of us understand what these natural products are or not. But the thing that I really want to highlight over here is that in 1960s and 70s, when all these anti-infective and basically anti-cancer therapies were being identified, natural product screening was the mainstay. And due to different reasons, which are, kind of highlighted in the red on this slide, natural product screening has taken a backseat. But again, in past couple of years, there have been new methods of culturing that have been identified, which has, kind of given, which have given a possibility of expanding the space, the chemical space that can be identified from natural product screening. There are improved methods of de-replication so that similar scaffolds that have already been identified do not get picked up again. And due to the developments in chromatography and spectrometry, their identification and the I would say structure characterization of the natural products have become relatively simpler as compared to what it was, couple of decades ago. And similarly, based on the development in metabolomics and that information available on the metabolite composition, it has accelerated the rate of de-replication of these structures. And of course, another way of kind of de-replication is that genome mining has helped in de-replication of these structures. So this is a classic example that I like to put in. Arylomycins, if you look at the lower half, arylomycins AST16 was one of the compounds that was identified as a class of natural products with weak activity and limited spectrum. And the focus was to convert this into a gram-negative activity. So the scientists at Genentech Roche worked for almost 10 years on this scaffold to identify G0775, which has a very potent activity too difficult to target the pathogen, especially the gram-negative, clinical isolates. And the compound is proceeding very well. So again, due to the development of various synthetic methodologies, understanding, that synthesis is not the only way forward, but also the biosynthetic engineering to produce analogs. Natural products with suboptimal pharmacological properties can be converted into compounds or analogs with superior pharmacological properties. This is again a classic example of tixobactin, which was the structure was elucidated by NMR and Morphe analysis, and this is a gram-positive antibiotic active against gram positive bacteria. So FBDD is again a complementary approach to HTS, and this shows a clear timeline of how FBDD has developed in last 3 to 4 decades, and the first breakthrough coming with the discovery of vemurafenib, again a kinase inhibitor that was had its roots in fragment-based screening to be FDA approved. And there are now, multiple compounds that have their roots in fragment-based discovery and followed by building up on those fragments to identify compounds with potent activity. And one classic example from fragment to marketed drug is the example of asciminib. So it was the first allostric BCR-able inhibitor to reach the clinic. And as you can see on the left, the discovery of how an inactive fragment, and because the activity is around 6 to 4 micromolar, how these inactive compounds were converted into by structure-based optimization into potent and clinical compounds AB001, which is first compound to reach the clinics. So I think this is a very recent publication, and actually this sums all that we have talked about, that there are technological advances plus how the way the compounds are screened. So this is a recent publication from JMed Chem, compound work done by Schrodinger, in which for MALT1, traditionally MALT1 orthosteric sites were being was being used for identification on inhibitors, and there were multiple MALT1 inhibitors in clinical trials failing due to some reason, either pharmacology or tox region, right? So how and the group at Schrodinger has identified SGR-1505 as allostric inhibitor, and you clearly see 8.2 billion compounds being triaged based on technologies like FEP, and only 129 compounds synthesized to generate SGR-1505, which is undergoing phase 1 clinical trial. So the kind of technologies they have used is FEP plus. Of course, these are their proprietary, Schrodinger's proprietary technologies. But yes, this gives an insight that how virtual screening using all these technologies can save time. And the important thing to keep in mind is that it took only 10 months to screen 8.2 billion compounds and synthesizing 129 compounds and then identifying 1505 to take it into phase 1 clinical trial. So not only with respect to the synthesis, I would say, but also with respect to the screens that are being used for screening the compounds. Again, a classic example, traditionally, rifamycins are known for their gram-positive activity, but no gram-negative activity in the normal media that is used for screening. But I think the group at Bioversus actually, screened the reframe library using a hypervirulent clinical isolate of A. baumannii and screened RPIM in presence of RPIM in serum, which mimics the bloodstream infections. And now the compound is in phase 3. So it gives, an insight into to think differently and to, use selected compound libraries to identify such kind of compounds. So I think, it is more to summarize that what we should screen and what we should be keeping in mind. Of course, one of the key factors that has to be kept in mind is resource allocation and timelines. whether it is known versus novel target, or what type of target it is, or what is the purpose of screening the library that we intend to screen, and whether the library is novel or, and should have minimum overlap with earlier screen compound collections. So I think the question to ponder on is, before anybody takes this screening library screening exercise, that are we screening smarter or we are just screening more? And if we say we are screening smarter, what kind of library we are choosing, and what do we intend to do with that library? How will we be screening? Are we using some, the traditional biochemical assays or phenotypic assays, or are we using different assays as we saw in case of, Acinetobacter screening, or, when we do different allostric site screening for MALT1. So I think with that, I'll finish my talk and happy to take any questions that you may have.